In this lecture about population genetics, I'm going to talk about how we track and decide whether microevolution is happening or not. That includes the Hardy-Weinberg equation, so if you're looking for information on that, this is the lecture you should watch. First, we just need to define a population, since remember, that's the smallest unit that can evolve, right? It's really important that you remember that individuals cannot evolve, and that's because they can't change their genes once they're born. And so evolution is at least microevolution, is the shifting of the genetic makeup of the population. So like in the finches that I just showed on that picture, it's like the beak depth or the beak shape. Does the population shift towards a larger beak shape or a smaller beak shape? Something like that. If you just think about it, of course the individual finch can't change its beak shape once it's born. So that's why an individual cannot evolve. And when we talk about population, we just mean same species in the same area interbreeding. The example they give here are two populations. They do have some overlap range, but the probability of breeding in that range is lower than breeding in their own range, and so we consider those two populations of the same species. You also need to know what a gene pool is. It's not a hard concept. Basically, you're taking all the organisms that live in a population, and you can think of having a big pot, and you just put all its genes in there. And we look at one gene at a time. So for example, in this gene pool, it's just a big bucket of all the X's. And each individual here has 2 copies of the X gene. That's because it's a diploid organism. It's what we're going to be talking about. And you can see there's 3 different alleles. Remember, those mean gene variants. I have another lecture that talks about how genetic variation arises as well. So in this case, we have the green number 1 variant, the red number 2 variant, and the blue, I'll call it number 3 variant. And we can see that in this population, there's a mix of those 3 types of alleles. If we had a gene pool where we only had red number 2 type alleles, that would be called having a fixed locus for the X gene. And sometimes we see that in populations, particularly in small populations where we have low genetic variation. And we actually have to worry about that in conservation biology, where more variation tends to allow the population to survive better. So when we're looking at the change in frequency of alleles, basically over time, do we get more or less green number 1 type allele? So if we looked over time, and the next time we looked at, or 3 or 4 times down the road, you had almost all the green number 1 allele, then you'd see a shift in the frequency of the alleles. The green number 1 would have gone up. And that's what we call microevolution, looking at those allele frequency changes. And if you remember any type of frequency, so if I ask you to calculate the frequency of women in a room, what would you do? You'd take the number of women divided by the total number of people in that room. So that's the same thing with these frequencies. For the allele frequency, we're just looking at the number of copies of the allele of interest in that population divided by the total number of copies of all the alleles in that population. So for example, we're looking at the number of green X1 alleles out of all the alleles that are present. So every number 1, 2, or 3 X gene. So if we wanted to calculate the allele frequency here of, let's say, what's the frequency of the X1, you take and count how many X1s there are. Out of how many total alleles? There are 30 individuals, each with 2 alleles, so that's 60 total alleles. And if you divide that out, you get 0.2. I like to put at least 2 decimals. And you'll notice here that you always need to write the 0 before the decimal point. It's a good practice to use for medicine and biology, because then I won't ever mistake that for a 20. You know, if you just put a tiny dot and it's not very clear, I might think that you wrote 20 instead of 0.2. But obviously if you have a 0 in front of it, you're not going to put that in front of a 20, so it's very clear to me that you have 0.2 there. So 0.2 is the frequency of the X1 allele in the population. So you could think of that sort of as 20% of the alleles are the X1 green allele. Genotype frequency is the same thing, but just for genotypes. So you're looking at the number of individuals of the genotype you care about divided by the total number of individuals. So in this case, if we wanted to know what's the frequency of X1, X1 individuals, we count how many of those there are. 1, 2, 3. And how many total individuals there are. There's 30 total individuals. So the frequency of the X1, X1 double green genotype is 0.1, which basically means 10% of the population is this homozygous for the X1 green allele. So what we can do is calculate the allele frequency from generation to generation, and as I mentioned, if it changes, that's the definition of microevolution. So here's how I like to sort of think about it. If you have 3 different genotypes in this case, we're going to say there's only 2 alleles at this locus, a big A and a little a allele. If you have more than 2 alleles, then you'd have additional combinations here, but we'll do this. So what genotypes? You can have a homozygous dominant, heterozygote, and a homozygous recessive. And then you want to say how many of each allele does the genotype donate to the pool. So the homozygous dominant genotype donates 2 big A alleles and 0 little a. The homozygous recessive genotype donates 0 big A alleles and 2 little a. And the heterozygote here donates 1 big A allele and 1 little a allele. And so these numbers are going to help us to calculate the total number of different types of alleles and therefore the allele frequencies. So there's what we just did here. If we want to calculate the frequency of the big A allele, it's this guy here, we actually have a term that we use for the frequency of that. It's going to be P. So whenever it's a lowercase p, whenever you see P, that always means the frequency of the dominant allele. If it's a situation where it's not complete dominance, then you designate an allele for P to stand for. This is just standard notation. This is the important one, is that big N means the number of individuals in a population. So we'll use that in our generalized equation. So how do we figure out the frequency of A? Well, we need the number of big A alleles divided by the number of total alleles. And how would we figure that out? Every big A individual donates 2 big A alleles. So you have 2 big A for every big A individual. So N, remember, that means the number of individuals of, in this case, of that type. But you also get big A alleles from the heterozygote. Each donates 1 from the total number of heterozygotes. Divided by, it's a diploid population, so every individual donates 2 alleles, whether they're big A or little a. So this equation is what P is. Now, of course, you can leave out this number 1 here, but I'm just going to write it in now so that you kind of get an idea of where these numbers are coming from. So what about the frequency of little a? We designate that as a lowercase q, and it's going to be the same thing, the number of little a's over the total number of alleles. And it's going to basically be a similar idea, but we're looking here. So the little a alleles come from 2 places. One place they come from is the homozygous recessive, so 2 alleles for every homozygous recessive individual, plus 1 allele for every heterozygote, divided by 2 alleles for every single individual in the population, and that would give us our Q. which is how we get this equation. So the frequency of the big A plus the frequency of the little a gives you the total gene pool. So this is one of our Hardy-Weinberg equations. You'll see what that means later, but you can keep that in mind. It's very logical. So what we've done so far is that we had the genotype numbers of the population, and we calculated allele frequencies. What we also can do is to calculate the genotype frequencies from the population data. So we just did allele frequencies, now we can do genotype. And so all that is the frequency of the genotype divided by the total number of individuals. First, let's calculate the observed genotype frequencies based off of this population data. So we're using these ideas here. So for the frequency of the big A, it's going to equal 90 individuals out of the total of 200. That will give us 0.45. Then we have the frequency of the heterozygote, which is going to equal 40 individuals out of 200, which equals 0.20. And last we have the frequency of the homozygous recessive, which is going to equal 70 divided by 200, which gives us 0.35. And hopefully that adds up to 100%, and it does. And so that is the distribution of genotypes in population 1, basically saying that 45% of the population is homozygous dominant, 35% is homozygous recessive, and 20% is heterozygous. Let's see the distribution in the population number 2. So the frequency of the homozygous dominant is 60 out of 200, which gives us 0.30. The frequency of the heterozygote equals 100 out of 200. And you'll notice actually this population is heavy on the heterozygotes. That's 0.50. And that's different, the heterozygotes, than the population number 1. So the frequency of the homozygous recessive is going to be 40 out of 200, which gives us 0.20. So again, these add up to 100 or 100%, which is good. So now you can see that the genotype distributions or genotype frequencies are different between the two populations, which just looking at the population data, you could see that that is the case. Let's calculate the observed allele frequencies for these two populations so we can compare the genetic makeup of them. So what's the proportion of big A's and little a's between the two populations? To figure out the frequencies, remember those are our P's and Q's. P equals 2 for every homozygous dominant individual, plus 1 for every heterozygote, divided by 2 alleles for all individuals, of which there are 200 here, and that's going to equal 0.55. So that's the frequency of the big A. The frequency of the little a in that same population is Q, which equals 2 alleles from every homozygous recessive plus 1 from every heterozygote. 2 alleles for all the individuals, of which there are 200, and that's going to equal 0.45. What about population number 2? All right, so P in this case, 2 alleles for every homozygous dominant plus 1 for every heterozygote, and 2 alleles for each individual, and that will equal 0.55. That's the frequency of big A. And the frequency of little a, Q, so 2 for every homozygous recessive plus 1 for every heterozygote divided by 2 alleles for every individual. You do that, equals 0.45. So in fact, if we look here, we have the same frequency of alleles in populations. Obviously they're distributed differently, but the allele frequency is actually the same. So that means the proportion of the gene pool that's big A and the proportion that's little a is equivalent in these populations. Now what we want to look at is whether that distribution of alleles is what we expect it to be. And if it's not, why would that be the case? We can actually calculate the expected frequencies based off the allele frequencies. And how we think of that is basically just probability. The probability of the offspring getting any particular allele is going to be basically the frequency of that allele. So if 0.8 is the frequency, so 80% of the alleles are big A, then we'd say that getting one big A is an 80% chance. So when you want two events to happen, then you have to multiply the frequencies together. So what that ends up being, this is the big A. So remember we said the frequency of big A is the little p. So now you need two of them. So you have the chance of getting one of them. That's frequency of big A times the chance of getting another one, and that gives you p squared. So now we need two of the little a gametes. We said the frequency of little a is q. So it's the same sort of thing here, but we need q to happen twice, to pick that twice. So that's going to be q squared. Now remember with the heterozygote, we actually have two ways to get our heterozygote. You can get the big A from mom and the little a from dad, or the little a from mom and the big A from dad, which just increases your probability of getting a heterozygote. So the big A frequency, right, is going to be your P. There's P. Your little a frequency is your Q, but because there's two ways to get it, we add a 2 in front of that. So in the end, the frequency of each genotype should all add up to 1. And so we get this equation. P squared, frequency of the big A, plus the frequency of the big A plus the frequency of the homozygous recessive little a equals 100% of the genotypes, which would be equal to 1. So this is our second equation. This is our genotype equation. And if you're having trouble remembering, all of these have 2 letters in it, just like a genotype would have 2 letters in it. An allele is like this, and a genotype is like that, right? So your equation with the 2 letters, that's going to be your genotypic equation. So now we have the allele frequencies from before. Now we need to see the expected genotype frequency based off of the allele frequencies. So p squared is going to be 0.55 squared, and that equals 0.3025. So that's the frequency, the expected frequency of the big A. And then we have 2pq, which is our heterozygous, so 2 times p times q, and that gives us 0.495. And then remember, that's the frequency of the heterozygote. And the frequency of the homozygous recessive, 0.45 squared, equals 0.2025. And so hopefully these add up to 1, so you can double check you did your math right. Now what we need to do is compare that expected frequency we just calculated to the observed frequencies. And then if they are the same, microevolution is not happening. If they're different, then we'd actually have to do some statistics to see if they're significantly different. But for our purposes, if they're different, we're going to say there's microevolution probably happening. You can see that population 1 does not match the expected at all. So we say that that one is undergoing microevolution, and particularly we can say that that one is not in what we call Hardy-Weinberg equilibrium, and we'll see that in a second. Number 2 population is pretty close to the expected, so if we did statistics, we'd see that that's not actually significant difference, and so that population would be in Hardy-Weinberg equilibrium. So let's look at that. Hardy-Weinberg equilibrium, it's the equations to describe a non-evolving population, and we've actually already looked at the equations, the P's and Q's. And they're derived from this Hardy-Weinberg Principle, which says that if a population is not evolving, then the allele frequencies will remain constant from generation to generation. So that means your P, every generation should stay the same, and your Q. And it also says that your genotype frequencies can be predicted from the allele frequencies. So basically, if you know what the P is and what the Q is, you should be able to calculate how those should be distributed as far as homozygous dominant, heterozygous, and homozygous recessive. And so how you figure that out is basically what we've just done. So you're going to test if the expected, which is the calculated, genotype frequencies match the actual genotype frequencies. And also you can, if you have generation to generation data, you can look at the allele frequencies and see if the P's and Q's change or not. In order for Hardy-Weinberg equilibrium to exist, there's 5 basic conditions that have to be in place. One is that the population size has to be technically infinitely large, but that's impossible. So I'll have a very large population size. And I'll just mark on here to recognize later, we're going to talk about this concept of genetic drift. So a large population size negates genetic drift influences. Random mating is going to negate sexual selection. This column is saying that you don't have consequences of genetic drift, you don't have consequences of sexual selection, no mutations occurring, no natural selection occurring, and no gene flow occurring. So when you see a population that is not in Hardy-Weinberg equilibrium, like the population number 1 that we saw, you have to figure out what's going on. And actually in this case, this is a population number 1 is butterflies, and there's actually been gene flow into the population. There's been a big migration event into that population. And so that's one of the reasons that those numbers are not distributed as we expect. It's also a relatively small population size, and so random differences make a bigger difference in a small population, and we'll look at that later in the genetic drift section, but just know that the small population size probably also influences the distribution not being expected. And even here, the distribution is not exactly as expected from the calculations, and that's probably due to the small population size as well. Let's look at a few problems. A study on blood types in a population found the genotypic distribution among the people sampled. Is this population in Hardy-Weinberg equilibrium? So to figure that out, we're going to have to compare the observed or calculated with the expected based off of the Hardy-Weinberg equation. And what do they give us? They give us genotypic distribution. The first we can show the observed frequencies, since that's easy, they're right here. Okay, so the frequency of the MM equals 1,101 people out of 3,100, and that equals 0.355. Okay. The frequency of the heterozygote, the MN genotype, 1,496 out of 3,100. That's going to equal 0.483. And the frequency of the NN genotype, that's 503 out of 3,100, and that equals 0.162. So those are the observed frequencies, and now we need to figure out the expected frequencies and compare the two. To figure out our expected genotype distributions, we need to use the alleles. So in the end, we're looking for p squared, 2pq, and q squared. And to calculate that, we'll need to find, obviously, p and q. So p equals, and we can use these equations again, in this case, 2 m alleles for every homozygous dominant plus 1 for every heterozygote divided by and 2 for all the individuals. That equals 0.596, so that's your P, and your Q is 2 N alleles for each homozygous N plus 1 for each heterozygote divided by 2 for every individual, and that equals 0.404. These add up to a 1, so that's a good sign. Okay, so now let's use those and calculate based off of those allele frequencies, P and Q, what we expect the genotypic distribution to be. P squared, which is what we expect the frequency of MM to be, is 0.596 squared, and that's equal to 0.355. Our 2PQ, which is the frequency, expected frequency of heterozygotes, so 2 times 0.596 times 0.404 equals 0.482. And finally, we have our expected frequency of the NN, that's Q squared, so 0.404 squared is 0.163. So we expect the distribution to be like that. And what do we see? Match, match, match. And the question asks you, is this population in Hardy-Weinberg equilibrium? And your answer would be yes. Let's look at another problem. Now, if you've paid attention so far, you've noticed that I have given you data for the heterozygote. Either I tell you how many heterozygotes there are in the population, or the heterozygote has a different phenotype so you can discern the difference between the homozygous dominant and the heterozygote. But what if the trait is completely dominant? And what that's going to mean, that's the important part, is that your big A and big A little a look the same. For example, in Mendel's pea plants, the purple allele was dominant to the white, so that meant homozygous dominant and heterozygote plants had purple flowers, and only the homozygous recessive had the white flowers. So what you'll have to do is to assume the population is in Hardy-Weinberg equilibrium in order to solve equations, or I'll tell you it's in Hardy-Weinberg equilibrium. All right, so here's another problem. In another study of human blood groups, it was found that among a population of 400 individuals, 230 were Rh plus and 170 were Rh minus. Assuming the trait is controlled by a dominant allele, as in Rh plus is big D, calculate the allele frequencies and how many Rh individuals would you expect to be heterozygous. They're asking us to calculate the frequencies of the two alleles, so in the end, we're going to need to find P and Q. They're also asking us how many are expected to be heterozygous. We could have also called those carriers in a complete dominant situation. So we need to solve 2PQ to find the expected frequency of heterozygotes. All right, what do we know? We know that there are 400 individuals and 230 were Rh plus, 170 were Rh minus. All right, so what we know are the phenotypes actually, and Rh plus, Rh minus. And let's look at the genotypes that go with those phenotypes. So Rh plus, what's the genotypes that give you that? Big D, and this is the key, big D, little d. How do you look Rh minus? The only way is to have little d alleles. And so this is the one that's going to be important to note, because if you can find the frequency of the little d, then you can find everything else. What is the frequency of the little d? That's our Q squared. When we think about frequencies, we're looking at the number of whatever we're interested in out of the entire population. And so for Q squared, the frequency of the little d, which gives you Rh minus, is right here. So 170 individuals out of the total of 400, that's frequency. So that equals 0.425. All right, so now we know what Q squared is. So what can we do with that? Well, what are they asking us for? Remember, they're asking us for PQ and 2PQ. So if we have Q squared, we can solve for Q. So how do you solve for Q from Q squared? If you take the square root of a square, that gives you Q. And so that means we want to take the square root of 0.425, which is going to equal 0.652. Meaning 65.2% of all the alleles in the gene pool are little d. Then we can solve for P because of this equation, which is just logic. 0.6 0.652 is Q, the rest of it must be P. So 1 minus 0.652 equals 0.348, and that equals your P. So that's the second part, and that's the frequency, remember, of your big D. Now they want to know what is the expected number of heterozygotes. So first we have to figure out the frequency. That's going to be our 2pq equals, now we have it, 2 times p times q, and that equals 0.454. That tells you the frequency of the heterozygous genotype, and they want to know how many individuals, and this is a population of 400, so we just take 400 and multiply it by the frequency, and that's going to give us 181.6, but we don't have points of people, so about 182 people are what we expect to be carriers or expect to be heterozygous. One more example. In a population, 96 out of 100 individuals exhibits the homozygous dominant phenotype for a completely dominant trait. What's the frequency of the recessive allele? So first, I think we'll figure out what they're asking us for. The frequency of the recessive allele. All right, the frequency of that recessive allele, which, okay, it's one letter, so that's going to be our Q. So they want us to solve for Q. So how are we going to get to Q? Well, let's see what they've given us. 96 out of 100, that's a frequency. Exhibit the homozygous dominant phenotype. Okay, so the dominant phenotype. So how do you get that dominant phenotype? Well, if it's a completely dominant trait, remember that means both the big A plus the big A little a will be giving you the dominant phenotype. So really 96 out of 100 is p squared plus 2pq, which does nothing for you. So instead you have to use like what's left over. Okay, so if 96 out of 100 individuals have the dominant, that leaves us with 4 out of 100 having the recessive phenotype. And how do you get the recessive phenotype? There's only one way, little a. So what we have here is the frequency of little a, which if you remember, that's two letters here, so that's q squared. All right, so q squared equals 0.04. I just divided 4 by 100. But what they're asking us for, remember again, we said, oh, they're asking us for q. So to take the square root of 0.04, and that's going to give you 0.2. So that's q. So the frequency of the recessive allele is 0.2, and actually I'll put 0.20. So that means 20% of the alleles in the gene pool are little a. And we could calculate any p's and q's that we need to from there, because now we have q and we can solve for everything. Now my question is, if this recessive allele is the mutant version of MC1R, remember that's the one in the rock pocket mice, and it results in a dark mouse coat color. If the population of 10,000 mice lives on the lava rock, what do you expect to happen to both allele frequencies in the next generation? So this is the frequency of little a, which is the mutant version, and it's going to result in dark fur. P is the frequency of the rest of the alleles, 0.8. You can also look at that equation. So frequency of big A, and that will result in the lighter phenotype. So the question is, if you have them living on the lava rock, what do you expect? So take a second and think about how you expect those allele frequencies to change and why. So what you should have come up with is that because the dark phenotype living on the dark lava rock is going to survive more often, you'd expect Q, the frequency of the little a allele, to increase. And because the light mice with the dominant allele are going to be picked off more easily by predators, you'd expect P, the frequency of the big A allele, to decrease. Now, how much that happens depends on how strong the selection is, but over time you'd expect to see the Q increase and the P to decrease. And that, of course, is due to natural selection. We would also say that the population is evolving because the P and the Q are not staying the same from generation to generation. Here are a few things I want you to think about with Hardy-Weinberg Principle. When we're looking at P's and Q's, right, we're just looking at one gene at a time. And so you might have one allele frequency changing for gene A, but other genes, gene B, C, D, might be at Hardy-Weinberg equilibrium. So you can't look at just the population as a whole. You'd be looking at the allele frequencies in a particular gene. So no actual biological populations are in Hardy-Weinberg equilibrium entirely. None of them meet all 5 criterion all the time, but we can use the Hardy-Weinberg equation to estimate changes in populations and expected frequencies. You'll also note here that some populations are very slow evolving, so you'd have to observe so many generations to be able to see the difference, that sometimes we don't have that data. And then in the end, just to note that really most human genes can be treated as Hardy-Weinberg, which is why we can do calculations with the equations. So this is how the human populations meet those 5 criterion. So genetic mutation rates low, mate selection is generally random. So let's say the allele we're looking at is a rare allele. That's a deleterious allele. You're only going to see natural selection select against that allele when it's homozygous recessive, which doesn't happen very often because most individuals have dominant alleles. Population is large when we look at human as a whole, and then in general, there are basically the same frequencies of alleles between most populations, and so migration, as in so people from different continents having children, that's not going to really affect the frequencies like you'd see in beetle populations from different continents, which are highly diverged. Why do we care about this Hardy-Weinberg equation? There are more reasons than this, but one important one is public health. Genetic counselors all the time are going to use this kind of data, as well as researchers, to calculate carrier frequencies, and that means heterozygotes, in particular populations, so they know whether to screen for certain types of genetic disorders, and they can study the population, understand why it's the way it is. So these are particular disorders that are very common in some populations but not others, so the carrier frequencies are really high. Cystic fibrosis in people of European descent, sickle cell disease in people of African descent, and like something like the BRCA 1 or 2 mutations in Eastern European Jews, or we can call that group the Ashkenazi Jew population. And so then you'd know that if someone is from of African descent, that you would probably look for sickle cell disease, and because that carrier frequency and thus the getting sickle cell disease is more common there. We actually look in natural populations of non-humans as well, and you can see the numbers and you can calculate whether that's Hardy-Weinberg equilibrium, and if it's not, that means the population is undergoing microevolution. And so you go and look at those 5 different characteristics and say which one or ones is being broken, and that can help you understand how populations are changing in the wild. And those are the basics of what I'm going to cover to calculate whether microevolution is occurring or not.